Hello everyone, welcome to today's live broadcast, Impulsivity and Compulsivity, Neural Substrates and Neuropsychiatric Implications. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We have a few important announcements before we begin. This presentation has been approved for a Category 1 Continuing Medical Education credit by Loma Linda University. If you are interested in obtaining CMEs after attending this presentation, please click on the Get Your Free CME CE Credits button in the bottom left corner of the event screen. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Trevor Robbins. Trevor Robbins was appointed in 1997 as the Professor of Cognitive Neuroscience at the University of Cambridge. He was elected to the Chair of Experimental Psychology and Head of Department at Cambridge from October 2002. He is also Director of the Behavioral and Clinical Neuroscience Institute, BCNI, jointly funded by the Medical Research Council and the Wellcome Trust. The mission of the BCNI is to interrelate basic and clinical research in psychiatry and neurology for such conditions as Parkinson's, Huntington's, and Alzheimer's diseases, frontal lobe injuries, schizophrenia, depression, drug addiction, and developmental syndromes such as attention deficit hyperactivity disorder. Trevor is a Fellow of the British Psychological Society, 1990, the Academy of Medical Sciences, 2000, and the Royal Society, 2005. In 2014, he was awarded a share of the Grete Lundberg Brain Prize. He was also given the American Psychological Association's Distinguished Contribution Award in 2011. Trevor has been president of the European Behavioral Pharmacology Society, 1992 to 1994, and he won that society's inaugural Distinguished Scientist Award in 2001. He was also president of the British Association of Psychopharmacology from 1996 to 1997. He has edited the journal Psychopharmacology since 1980 and joined the editorial board of Science in January 2003. He has been a member of the Medical Research Council UK and chaired the Neuroscience and Mental Health Board from 1995 until 1999. He has been included on a list of the 100 most cited neuroscientists by ISI, has published over 600 full papers in scientific journals, and has co-edited 7 books. I will now turn it over to Dr. Robbins for his presentation.
Hello, so this is Trevor Robbins, University of Cambridge. I'm going to be talking today about 2 concepts which I think will be of great importance in psychiatry in the future: impulsivity and compulsivity. So let's see what we say about impulsivity. What do we mean by this concept? Well, there it is in the middle, and here's a definition. Actions which are poorly conceived, prematurely expressed. In other words, behaviors which are blurted out inappropriately too soon. Unduly risky, inappropriate to the situation, and often result in undesirable consequences. Now, the prototypical disorder in mental health for impulsivity may be ADHD, attention deficit hyperactivity disorder. But it also includes substance abuse disorders, drug addiction, mania, and obviously personality disorders such as antisocial behavior. Now, in psychological terms, that definition I've given you covers quite a lot of stuff. So the type of mechanisms we're discussing probably include motor disinhibition, the loss of inhibition over your motor behaviors, intolerance to delays, impatience, especially when you're waiting for rewards, and also a lack of consideration or reflection when you're making important decisions, making the decision too soon on the basis of not enough information. Now, is this a unitary thing, this impulsivity, or can it be divided into different types? I think the latter, but we'll get to that later in the talk. Now, in terms of the brain, impulsivity probably involves some of the systems that are depicted in this slide. So on the right bottom hand corner, you can see a diagram of the human reward system with its frontal lobe components and also the nucleus accumbens, which is there. Now, depicted on the left-hand side is this circuit schematically. It's a hot loop, so it's shown in red. It's a reward circuit. There are other parallel circuits involving the frontal lobe, but we're interested in this one. This circuit's very interested in anticipating rewards, and this circuit turns out to be pretty important for impulsivity. Now, these circuits are all modulated by neurotransmitters coming from forebrain and midbrain structures. They include the dopamine and serotonin and noradrenaline systems, and they innervate different levels of this loop, and they're also implicated in this behavior. Now, we know a lot about this system because originally it was shown in animal work that this system is involved probably in the rewarding effects of drugs as well as natural rewards such as food. And in particular, the dopamine system plays an important role in this. So, for example, drugs such as amphetamine and cocaine, which work through the dopamine system, have their effects mediated in this circuit. Now, this work has transferred or translated perfectly to human work through the work of people like Nora Volkow, who's related to Leon Trotsky, the Russian revolutionary. And what Nora found is that in chronic drug abusers, such as cocaine or methamphetamine or alcohol abusers, they had reduced dopamine receptor binding in the stratum, in the human stratum. And you can see that in the diagram. You can see these levels of dopamine receptors less than in normal subjects. You see they shine less brightly. But one big question to ask is whether this is a cause or an effect of drug abuse. So, for example, you might think that this has resulted through chronic years of drug abuse, but on the other hand, it might have been there before they abused the drugs and actually contributed to the drug abuse. So how do you work out a cause or effect in this situation? There's a similar problem with behavior. So all of these studies have shown that drug abusers are impulsive. They show this premature risky behavior, whether it's stimulant abusers, opiate abusers, alcoholics, or smokers, nicotine abusers. But again, there's an issue. Were they like this before they took drugs, or did the drugs make them impulsive and impair their decision making? It's just not so clear. So there are ways, however, of addressing this tricky issue of cause and effect. Now, humans, when they measure impulsivity, use the so-called Barrett scale a lot, the Barrett impulsiveness scale. And this has questions in it like, I don't pay attention, I'm self-controlled, and they generally say no. I change jobs, they would say yes if they're impulsive. They're restless at lectures, impulsive people, so they jump around a lot. They're motor disinhibited. So this is how one measures impulsivity in terms of self-report, introspection. But there are objective ways of measuring impulsivity as well, and fortunately we can use these methods in rodents, in rats, as well as in humans. So these methods include measures of patience when choosing rewards. This is called delayed gratification. They include a test called the stop signal reaction time test, which is a test of motor inhibition. And they include attentional tests, like the test you see in the lower left-hand panel here, of a rat, in this case, trying to detect brief flashes of light which predict food. And the rat has to nose poke in these apertures to receive the rewards of food, which are collected here, and then he has to turn round and attend to the next cue. Now, the interesting problem for the rat is that it has to wait until the cue comes on before he must respond. And there is a proportion of rats in the lister population. This is a lister hooded rat called Zippy. There's a proportion of these rats who show very high levels of impulsivity, as you can see here. They're out with the normal distribution. They have a consistent tendency to respond too soon. Now this breeds true when you do behavioural genetics experiments, and indeed Jeff Daly in our group has shown that the genes involved are on chromosome 1 in the rat. Now this is a very important trait, therefore, this premature responding. And it's been shown by Jeff Daly and others that rats who show this high level of impulsivity, when subsequently allowed to self-administer drugs such as cocaine or nicotine intravenously, and they choose to do this by hitting a lever in this special box. They hit that lever there, and it produces a shot of cocaine in the vein. Now, if this schedule is allowed to develop so that the rats can respond for long periods to the drug, intake of the drug may escalate. And this escalation occurs especially in high impulsive rats for cocaine, and also for nicotine if they're allowed access to nicotine, and interestingly also for sucrose rewards. So these high impulsive rats are very sensitive, as it were, to the rewarding effects of drugs of abuse. And that's a very interesting observation, because it suggests to us that, well, impulsivity is not necessarily just caused by the drugs, it occurs beforehand and may even contribute to the risky behaviour that is drug seeking. Now additionally, Jeff Daly also found, intriguingly, that in the ventral stratum of these rats there was lower dopamine receptor binding, just as had been shown, here you see, previously in humans. And this low receptor binding is also correlated with the degree of impulsivity, as you see in this inset here. So these high impulsive rats have low dopamine receptors, just as human drug abusers do. And of course, these receptors are measured before the animals took the drugs, and so they were part of the trait of impulsivity, which may contribute to drug abuse. Now, other studies have confirmed that these receptors are reduced in the so-called shell region of the nucleus accumbens. That's shown in the middle panel at the top. And also other molecular changes in this region suggest to us that actually the dopamine transporter is reduced as well, and suggest that the dysregulation of dopamine in this region is contributing to the impulsive behaviour. Now, the adjacent part of the nucleus accumbens is called the core region, and the core region of the nucleus accumbens also contributes to this impulsivity. So here you see the reduction in grey matter in the nucleus accumbens core in a section of the rat brain measured with magnetic resonance imaging, and the graph below shows you how well this correlates with the tendency to impulsivity. So the less the grey matter, the more the impulsivity. Now that grey matter is probably so-called medium spiny cells, which use GABA as a transmitter. And indeed, as you see on the right-hand panel, these rats have less of the enzyme which makes GABA, GAD as it's called. And they also have less synaptic proteins in this region. Intriguingly, in normal rats, if you impair GABA with antisense, if you impair this enzyme, as you see in this bottom right-hand panel, they show increased impulsivity. So what all this adds up to is that the core region of the nucleus accumbens is also implicated in impulsivity. And we've seen this in another test where rats show very steep reward discounting if they have lesions of the core region. So in this bottom right-hand corner, you see the lesion of the nucleus accumbens here, And this leads to an increased tendency to take the smaller of two rewards if the smaller reward is presented ahead of time, before the large reward. And you can see this discounting function on the left, the steeper discounting, here we go, of the high impulsive rats. So they're impulsive according to two criteria, premature responding and impatience for reward. And it all devolves on the nucleus accumbens. So this is our neural network of the nucleus accumbens, and you can see the nucleus accumbens core and shell regions both contribute to this behaviour. But additionally, there are top-down controls on these structures, as you see on the right-hand side, and these are particularly from the frontal lobes, as you see here, controlling the output of these structures as well. On the left-hand side, you see not only the dopamine system projecting to the nucleus accumbens, but also the serotonin system and the noradrenaline system, which goes to the shell. And they all play a role in regulating impulsivity. And that's also seen from studies of people with ADHD, where drugs such as Ritalin, affecting dopamine and noradrenaline, are used to control impulsive behaviours. But what does all this mean for drug abuse, human drug abuse? Is impulsivity really what we call an endo phenotype? Is it core to the condition, and does it contribute causally to the problems of drug addicts? Well, here's some translational experiment where we used something very similar to the rodent attentional task for humans. It's a 4-choice task. The human holds down a button, and you can see where that happens here. And when the visual stimulus occurs, you release the button and touch the screen, and you get a reward for that. Now, the subjects are encouraged to respond as quickly as possible, so they do make mistakes and they do release the button prematurely. But guess what? Just as in rodents, we find stimulant abusers and recreational cannabis abusers both make more premature responses. They both release the button too soon. So there you have a real translation from the rodent to the human. It shows that that rodent work makes some kind of sense. Now, another impulsivity task is the stop task. And this is a fun task. You're supposed to respond as quick as you possibly can, either to the right or to the left, activated by a computer cue. So these come up very quickly. But on some of the trials, a beep sounds. And when that beep sounds, you mustn't respond. You have to cancel your response. And it's possible using this task to measure not only how quick you can respond, but also how quick you can stop. You can inhibit a response. So imagine an ADHD child might blurt out something. You might want them to stop that response, and this is a kind of lab measure of that. And indeed, with ADHD, these are adult patients, if they're treated with Ritalin, which is the drug treatment for ADHD, it does improve their stop signal reaction time. Here it's very slow compared to controls. Here's the controls, you can see it's slower. Under Ritalin, they're able to stop more easily, so they can control their impulsive behaviours. Okay, now this structure, this network, is the network which controls stopping. It's a bit different from the waiting circuit. It's a stopping circuit, and it involves similarly structures in the basal ganglia, in the stratum, but they're in the caudate putamen and not in the nucleus accumbens. They're also controlled by some top-down structures from the frontal lobes, notably the inferior frontal cortex, and they're also modulated by these ascending chemical systems, dopamine, serotonin, and noradrenaline, on the left-hand side of the slide. Okay, now intriguingly, we can use some of this evidence when studying addiction. So in the top left hand of the slide, you can see the relationship between damage to this area of the frontal cortex. It's on the right side and it's inferior, and the stop signal reaction time. Look at this amazing relationship. The slower you are, the more damage patients with damage in that area have in that particular zone. So this zone is key for stopping. Intriguingly, this zone is also compromised in stimulant abusers. And Cowan-Ursher has shown this in this study of the stop signal reaction time task, where she showed that not only do stimulant abusers show slow stop signal reaction times compared to normal there, so do their siblings who don't take drugs but are family members and therefore genetically related. So it's intriguing that in both the siblings and the stimulant abusers who are bad at inhibiting their responding, that this correlates with their white matter as measured by diffusion tensor imaging in this region of the frontal lobes. And you can see this fantastic relationship between the stoppability and the loss of innervation of the frontal lobes in the stimulant abusers and their siblings. Note this very interesting control group. These are recreational drug users. They take a lot of stimulants, such as cocaine, but they don't seem to develop addiction per se. They don't develop compulsive addiction-like behaviours, and nor do they show this impulsivity endophenotype. So it looks like impulsivity may be linked to the tendency to become stimulant dependent or stimulant addicted. Now we've got a similar story when we studied healthy adolescents prospectively, because this is another way to get at cause and effect. So in this study in Europe, 2,000 14-year-olds were recruited from schools, normal 14-year-olds, and given a whole battery of tests, clinical tests, neuropsychology tests, brain structure, functional brain imaging, the whole lot. And I'm going to show you a snapshot of the data on the stop task, because these are sections of the brains. This is a summary of those networks which were activated during successful stopping, and the ability to successfully inhibit a response. And you can see, intriguingly, these frontal areas, orbitofrontal here and here, right inferior frontal, that area I mentioned earlier, basal ganglia here, this red areas, and also structures such as the supplementary motor area, which probably delivers the inhibition of the response. Now, as I said here, different networks are associated with different aspects of impulse control, but adolescents reporting drug use, that's alcohol, nicotine, or both of these substances, had reduced activity in these lateral green areas. Intriguingly, those adolescents additionally reporting illicit drug use had initially increased activity in this right inferior frontal area. So there's something abnormal going on there. And we know later on in the course of drug abuse, this area becomes hypoactive, less active. So it may be that they're trying to control their drug taking, but eventually can't do it. So here's two bits of evidence, therefore, to show you that these relationships between impulsivity and stimulant drug addiction appear to be there. But how does this factor into compulsivity? What is compulsivity? Is it different from impulsivity? It's a very similar definition, but it is different. We would argue that it's actions which persist inappropriate to the situation. They perseverate, they continue. They have no obvious relationship, however, to goal-directed behaviour, and they often result again in bad consequences. Now, these impulsivity could be due to motor disinhibition. You get these syndromes called stereotypy, for example, in stimulant abusers who perform the same actions over and over again. It could be enhanced habit learning. This is one of our favourite hypotheses. As behaviour becomes more automatic, we think that the purpose of the behaviour is neglected and the behaviour becomes an end unto itself. Persistent behaviour is shown when extinction is studied, when the reward is taken away or the contingencies change, and also just when you think in a very rigid way, when you have cognitive rigidity. Now, what do these traits contribute? Substance abuse is defined nowadays almost by the new DSM-5 diagnostic system as compulsive drug use. Eating disorders can be compulsive. We all know about gambling and compulsive gambling. And prototypically, OCD, obsessive compulsive disorder, is the ultimate compulsive behaviour. Now, is there something called compulsivity which covers all of these conditions? Is there something in common to them? Well, let's consider this. First of all, this brilliant quote from the great clinician Chuck O'Brien. It's a great addiction clinician. I'll read it to you. At some point after continued repetition of voluntary drug taking, the drug user loses the voluntary ability to control its use. At that point, the drug user becomes, where are we going here? The drug user becomes, I'm sorry, I'm just losing a bit of control here. I'll be with you in a second. Just went the wrong direction. Yeah, at that point the drug misuser becomes drug addicted, and there is a compulsive, often overwhelming involuntary aspect to continuing drug use and to relapse after a period of abstinence. So this is clearly arguing that there's a transition from goal-directed or voluntary behaviour to automatic, we would argue, habit-based behaviours. Do we see this in animals? Well, we can measure compulsive behaviour if we introduce punishments for drug taking. So, for example, if the rat is given a mild electric shock for continuing to self-administer cocaine, will they continue to do so? Normal rats will not, and that's shown with this little green histogram here. However, the high impulsive rats I mentioned earlier generally continue to respond. You can see this huge effect here. They continue to respond even in the face of receiving the adverse consequence of electric shock. So, although it's quite a small proportion of normal rats, it's those rats who develop compulsive tendencies. There are enormous individual differences here. And that's true also of stimulant abusers as humans. It's been estimated that only about 17% of cocaine abusers actually become cocaine addicts. Okay, so a hypothesis that Barry Everett and I have suggested is this link between impulsivity, mediated by the nucleus accumbens or the ventral stratum that you see here, turns into, with progressive drug use, habit-based behaviours involving the progressive involvement of the dorsal stratum. And also this leads to relapse susceptibility, as you see here. So the neural basis of this is almost certainly this wiring diagram shown here of the basal ganglia. This has been illuminated beautifully by Suzanne Haber. So what she's shown is that there's a cascade of circuits going from the nucleus accumbens back to the source of the dopamine system, and thence up to the next area of the stratum, before coming back to the next controlling dopamine system, which projects to the next inline part of the stratum, and so on, until you reach the putamen of the stratum. Now, the interesting point here is that the putamen has been linked to habit-based behaviours, stimulus-response learning, where the goal is no longer as important as the skill or the action. Now, how can we provide some evidence for this? Well, this is a habit-based behaviour of rats responding a lot on a lever to produce cocaine infusions occasionally, as you see here, after 15 minutes. The behaviour is held together by these stimulus-response connections, if you like, which occur every 10 responses during the interval. If the stimuli, the yellow light, as you see in this diagram, is not present, then the behaviour collapses. Now, it's been shown by my colleague Barry Everett and Luc van der Schuren that if you block the dopamine receptors in this dorsal striatal region, you get a dose-dependent reduction of self-administration of cocaine, suggesting that the control of the behaviour has been devolved now to this dorsal striatal structure. By contrast, the nucleus accumbens is no longer important. Dopamine blockade there does not block the goal-seeking behaviour. So the idea, therefore, is that the control over this behaviour has devolved from the ventral stratum, the nucleus accumbens, to the dorsal stratum. And that's why it's susceptible, only after a lot of training, to dopamine receptor blockade there. Now we come back to this theoretical scheme that drug addiction really is an example of instrumental conditioning. And what I mean by that is action consequence learning or action outcome learning. So all this means simply is that if you perform a particular response in a particular environment, and that leads to a goal such as a reward or what we call a reinforcer in the form of a drug or even a drug-related cue, a stimulus predicting the drug, this all represents goal-directed behaviour, where you've got control over the environment, your response is producing an outcome. And this, we think, represents drug use and misuse, abuse. But habit learning occurs with incredible repetition. Gradually, the stimulus-response contingency takes over, and the reward is no longer as important. So the drug abuser is performing the behaviours, but the drug somehow is no longer as important as it used to be. The behaviour itself has become an end in its own right, if you like. And this point, we suggest the habit is a building block for more compulsive forms of behaviour. Now, we know a lot about the neural basis of these two systems, the goal-directed system and the habit system. And guess what? They're in parallel circuits in the basal ganglia. So this circuit has been shown with food-related behaviours in humans to consist of the caudate nucleus here. This is an fMRI study, a brain activation study. The caudate nucleus and that part of the orbitofrontal cortex in the middle between the eyes that you see here. That's the goal-directed system. The habit learning system, however, is mediated by this circuit. Now, how do we test goal-directed versus habit learning? The way we do that is we devalue the goal. So for rats and humans, we just give them lots of the flavour in advance so that it's no longer as exciting as it used to be. Or maybe we poison the food a bit, make the rat a little ill or something like that. And then see whether the animal or the human continues to respond for this devalued goal. Now, if it's a habit, they should do, because the behaviours become automatic. But if it's still goal-directed, they should stop responding because the goal has been devalued. That's the way the logic works. Okay, so let's see if we can apply some of these tests to impulsivity, but more especially compulsivity. Habits as the basis of compulsive behaviour. So first of all, OCD. So here's some examples of OCD, quite dramatic ones. This is hoarding. I joke that this sometimes represents fellows at Cambridge, their offices, but that's not really true. Also at Cambridge, we're very particular about having low-cut graphs, and you can see this perfectionism. That's another aspect of OCD. Hand washing, because of contamination fears, is another really important symptom of OCD. And even famous people complain of OCD. For example, here Beck's, but many other cases I could mention. And these are very severe symptoms in some cases. They may prevent someone from actually working or even going out of their house during the day because they get caught up with their rituals, which they perform for hours and hours and hours. Often they say to relieve anxiety, but we have a view on that. Okay, well, there are interesting connections between stimulant drug abuse, stimulant drug addiction, and OCD. And here's a study which seems to show that. In this study, we used what's called resting state magnetic resonance imaging with connectivity measures to look at the wiring of 50 different areas in the cortex of the human brain. So here you can see two red areas highlighted on the left-hand side. These areas turn out to be in the resting state, particularly disconnected, both in OCD and also in stimulant drug abuse. These two areas. Intriguing that it's the same two areas in these two very different, or apparently different, conditions. And you can see on the right the quantification of this, the reduced connectivity of these areas with other areas of the cortex. That's interesting. But what's even more interesting is if you measure compulsivity with a drug scale called OPDUS or an obsessive compulsive scale called YBOX, this disconnectivity correlates with the degree of compulsive behavior, either for drugs, which often means doing drugs rather than necessarily liking drugs, and also for OCD, the level of obsessive compulsive symptoms. In both cases, there's a relationship between the disconnectivity of the frontal lobes and the propensity to perform these compulsive behaviors. That gives me some optimism that we're dealing with a similar kind of thing. There is something called compulsivity which affects both groups. Now, further evidence that they're related. Here's some more work from Karen Urscher showing, if you look at the central panel, that compulsive disorders, obsessive compulsive behaviors, are quite prevalent in stimulant drug abuse and also, as you see here, in their siblings compared to controls and also recreational drug users. Very interesting. Recreational drug users tend to be sensation seekers, but the siblings of compulsive drug users do not tend to be sensation seekers. So we think these are very different risks, if you like, for drug abuse. And on the left-hand side here, you see the data you saw earlier, showing the link perhaps between impulsivity and compulsivity in the stimulant abusers and also in their siblings. Okay, OCD and habits. Can we test whether they have more habits than normal? So our hypothesis has been that OCD patients have a disrupted balance between habit learning, what to do in the context of a particular stimulus, and goal-directed behaviors, learning about consequences, learning that if I do this, then that will happen. And our hypothesis is that this bias to habitual behavior may underlie their drive to perform compulsions. And OCD is a kind of behavioral addiction as a consequence of that. Now, maybe this bias is enhanced by stress and anxiety, and that's why the habit-based systems become compulsive in their performance. But that's something we'll address in future research. Having addressed the issue of the balance comes from 3 tests of habit learning for humans that we've used. We've used one test of aversive habit learning, which is obviously very relevant in OCD, but I won't have time to talk about it this afternoon. We've used another test of habit learning versus goal-directed learning, which is an ingenious test based on kind of fruit machines, looking at the balance between what's called goal-directed behavior, model-based behavior, and model-free learning, which is sometimes represented as habit learning. So we've done that, but again, I don't have time to describe that. I'm going to focus on the fabulous fruit game. So the fabulous fruit game is a human learning task where you can study the balance between goal-directed behavior and habits. And you can do this by devaluing the goals or the rewards that are obtained in the task. How is this done? Well, in this task, what happens is you have to learn the association between a stimulus on a box, which is in the form of a fruit, and a button, left or right button, and knowing which button to press in the presence of that fruit to earn a reward, which may be another fruit, which earns you points. So let me describe this in a little more detail. Here's the stimulus, it's bananas. You've got to decide whether to press the left or the right button. The outcome is a cherry, which would earn you points if you get it right. And what we've found is that if you have to learn a whole lot of these associations, Patients with OCD and healthy volunteers are just the same as one another. They show normal learning. However, the differences come when you devalue the rewards. Now, how do you do this? Well, you just simply tell the subject that bananas are no longer valuable. They don't earn you points, whereas cherries are still good, for example. And grapes, as you see here, are devalued. And then you give them a test. You show them the stimuli on the outside of the boxes, which of course are linked to these rewards, and the subject has to decide whether to respond to the stimuli or not. Now, if they respond automatically to the stimuli, it's going to be a habit, and we call this a slip of action, as you see here. This is a test of habit learning. And the more slips you make, the more habit-bound you are. And this is the case for OCD patients, as you see here in blue. So they are more prone to make slips of action. They are more prone to show evidence of automatic stimulus response habit learning, irrespective of the devalued goal. By contrast, and intriguingly I should say, this correlates with their symptoms quite highly. Doesn't correlate with their anxiety or depression, but correlates quite remarkably highly with their symptoms. So this lab test is telling us something, perhaps, about their symptoms. Now, the other side of the coin is, can they remember the consequences of their actions? Can they remember which button to press to get the reward, left or right? And here you see that they're less good at that. Intriguingly, they show impaired action outcome learning. So improved habits, impaired action outcome, balance shifts towards habit learning. And moreover, if they're quizzed on the associations, just with a questionnaire where they have to simply ring things in order to say which is the right response for getting which fruit reward, they're also very bad at that. And you can see that here. They're very bad at subjectively remembering the consequences of their actions. They are more habit prone, they are more biased to habits. And we see this for aversive as well as appetitive habits. So we think, therefore, that habits may be a building block of their compulsive behaviours. Now, what about addiction? Can we show that this is relevant for addiction? Well, there is evidence from Sanne de Wit, who invented this task, she's in Amsterdam, that if you look in the brains, even of normal subjects, there's correlations between white matter connecting the putamen of the basal ganglia, here, with the motor cortex, and the slips of action test. Whereas there's an opposite relationship for the goal-directed system and the slips of action test. So in other words, it's possible to see visually the strength of the connectivity in the brain associated with the proneness to habits and possibly therefore the proneness to compulsive behaviour. Now, we haven't applied this directly to drug abusers yet, but we've, in collaboration with Sanne de Wit and others, we've used a similar task of the fabulous fruit game to get at habit learning in alcoholics. And this, in this study, by Stewards and others, which I was fortunate enough to collaborate on, we adapted the fabulous fruit game in the following way. On the left-hand side, you can see some of the associations that people have to learn. If one fruit, then this response gives you an apple. So the way you remember the action is that this fruit reminds you of the apple, and the apple reminds you to press the right lever. That's how you do it. Now, this task is particularly easy, obviously, if the fruits are the same. There's no confusion here whatsoever. If cherries, then a right response will give you cherries, and similarly for oranges. But in this special case here, you see a problem if you use the goal-directed system, because, if we go back, sorry again, because you can see here that you can be confused. If strawberry, then pear says press right button, But in this case, you see the pair again, and although it reminds you of a strawberry reward which says press left, the pair might confuse you and say press right. Actually, the only way to do this task is to use the habit system and to learn a simple rule, which I show here. If strawberry, press right. If pear, press left. Don't try and remember the consequences of the actions, just do the habits. That's the only way to do that task. Now, taking advantage of that, we looked at a group of alcoholic patients. Here is the study involved. And we showed that there were intriguing differences between healthy volunteers and this group of alcohol-dependent subjects. First of all, the healthy controls had increased activity in these tests in the goal-directed system, in the caudate nucleus, you can see here, and also in the orbitofrontal cortex, rather ventral medial area there. So the goal-directed system is working less well in the alcoholic-dependent group. By contrast, and this is the interesting finding, the putamen here is more active in the incongruent task in the alcoholic disordered group, much more so than the healthy controls. So in essence, this suggests that their habit system is more active in this task, and they're making more use of it. So that's indirect evidence that the alcoholic abusers prefer to use the habit system. Now, maybe it was always that way and their relatives showed the same effect. We haven't tested that yet. Maybe the alcohol has helped to induce this drift to the habit system. Maybe anxiety or stress has made them more dependent on this system. But whatever it is, the habit system may contribute certainly to their drug-seeking compulsive behaviours. Okay, so I'd like to wind up now, finally, and discuss how these ideas work in the context of the brain. Here you see a schematic of the same brain circuits we've been talking about. These are the different loops. They're represented in this case, however, horizontally rather than in parallel longitudinally. So I started off by talking about an impulsive waiting circuit in yellow, which involves the nucleus accumbens, this structure here, and certain areas of the frontal cortex. I also talked about a stopping circuit, which is in parallel, which involved the caudate nucleus and structures in the frontal lobe, in humans, the inferior frontal cortex. So these systems very importantly control impulsivity. By contrast, these other systems, going down the slide, are more involved in compulsive behaviour. So whereas this is all to do with anticipation of reward, this is all to do with what happens after the end of a response sequence when reward has been delivered. Now there is evidence, which I haven't described, that this system is very much involved in flexibility of goals. It's certainly involved, as I showed you, in goal-directed behaviour.
Unfortunately, we are out of time. I want to thank the audience for their interest, for participating in today's event, and our speaker, Dr. Trevor Robbins, for his informative presentation. Today's webcast will be available for on-demand viewing for several months following. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.